Okay, hello everyone. My name is Quentin Leclerc. I'm a final year PhD student at the London School of Hygiene and Tropical Medicine and at St. George's University of London. And today I'm going to be telling you about modeling the dual nature of bacteriophage in the context of AMR. So first of all, what are bacteriophage or phage or phage? So these are the most abundant organisms on the planet and natural predators of bacteria. So for this reason, these bacterial viruses are currently being considered as part of phage therapy, which is a potential solution to tackle AMR and relies on using these phage as antimicrobial agents. However, in reality, phage have a dual nature because they're also major drivers of bacterial evolution through transduction. So this type of horizontal gene transfer has been shown to lead to significant AMR evolution. The problem is we currently don't have a good understanding of the dynamics of transduction of AMR. We know that it happens, but in terms of the rates, in terms of how this varies depending on the number of bacteria in phage, we don't know yet. So the three previous studies which have attempted to model this had to rely heavily on assumptions and previous data from various sources. So here our aim is to try to investigate the phage bacteria dynamics, including transduction, using an interdisciplinary framework. So simultaneously generating lab data and also a mathematical model in parallel to really have to rely on the minimum number of assumptions and really understand what's going on here. So just to flag, all these results are available in a preprint, so please refer to this for more information on the methods and results. Okay, let's start with a bit of biology to make sure we're all on the same page. So what is transduction? So full disclaimer, in case you're familiar with transduction, I'll only be talking about generalized transduction, not the other mechanism of transduction, which is specialized transduction. Okay, so what happens when phage and bacteria are together in an environment? Well, typically, lytic phage, in gold here, will infect a bacteria, inject their phage DNA, replicate using the bacterial machinery, and create new phage, and then burst the bacteria to release the phage back into the environment. So this lytic cycle is really what's being considered for phage therapy to get rid of the bacteria. However, in that process, a mistake can happen at the packaging and assembly step. So what can happen is instead of the phage DNA, a bit of bacterial DNA can be packaged into a new phage. So in that case here, an AMR gene. And that transducing phage carrying the AMR gene will be released alongside the others when the bacteria bursts and can then inject this AMR gene in another bacteria. So that's horizontal gene transfer via generalized transduction. So it's these two things, so the lytic cycle and generalized transduction, which form the dual nature of phage that I'm really interested in looking at. And these are the dynamics I'm trying to disentangle here. So that's for the biology. Now, the first thing I need to look at all this is data. So what kind of data do I get? So I work in the lab to get this, and I work with Staphylococcus aureus bacteria, which is the perfect model organism for this, because transduction is the dominant horizontal gene transfer mechanism in that bacteria. So I do cultures of phage and bacteria. So I work with two single resistant strains of bacteria resistant to either erythromycin or tetracycline. And then I also add in some lytic phage in the mix and grow these tubes over 24 hours. And based on the biology of transduction, I know that eventually in that tube, I'm going to get transducing phage that are going to appear that will carry either the erythromycin or the tetracycline resistance gene. And these transducing phage, by then interacting with the bacteria again, will eventually lead to double resistant bacteria, carrying both the eri and tet resistance genes. Okay, so the advantage of that system is it's actually straightforward. I know all the organisms that I have in there. I can count all the bacteria. I can count the lytic phage. The problem is I cannot count the transducing phage. These are effectively invisible. I don't have an easy way to count them. So this will tell me the numbers of things I have, but it doesn't tell me how all these components are interacting together. So that's where the model helps. So I've built a model in parallel of this, which contains all the components I've just described. So the three bacterial strains and the phage, but all the interactions between them as well. So the bacteria replicating, the phage replicating as well by exploding bacteria, and this process sometimes creating transducing phage, which can then infect the other bacterial strain to lead to double-resistant bacteria. All right, so now I've got my system where I've got the lab data and the model effectively showing the same thing. Hopefully I can make both of them talk to each other and reveal all the underlying dynamics in my system. So first of all, what does the lab data look like then? So I grew bacteria and phage over 24 hours, counting them hourly between 0 and 8 and 16 24 hours, and starting the single resistant bacterial strains at around 10 power 4 bacteria per mil. And the phage, I either started at around 10 power 3, 10 power 4, or 10 power 5 per mil. Okay, so first thing we can see from this is that I'm detecting double-resistant bacteria as early as 7 hours, and this tells me for sure that transduction has occurred in my system, because these cannot appear originally here by any other means than by transduction. So that's the first thing. Second thing which is interesting about this is that the bacterial and phage dynamics seem to vary depending on the starting number of phage. So what this means is, if you look at the 10 power 3 and 10 power 4 datasets, the bacteria grow for about 8 hours, then they reach their stationary phase. And interestingly, the phage also stop increasing after 8 hours, which is somewhat unexpected, because we'd expect the phage to just keep killing all the bacteria. As for the 10 power 5 dataset, however, in the later time points, the phage are still increasing, and we're starting to see a bit of a decrease in the bacterial numbers. So that's the data, that's what we can see, but the invisible dynamics, we have to rely on the model to figure them out. So I fitted the model structure that I showed you before using MCMC methods and varying the types of interactions between the phage and bacteria until I was able to get something which could replicate these trends. And there was only one model which was able to do this, and this is what the output looks like. So that's all the dashed lines. So again, it's a model that is able to replicate these trends because, as you see here, it's correctly predicting that the phage numbers stabilize in these two datasets after 8 hours. And here it's also capturing that increase in the later hour in phage numbers. It's also generating double-resistant bacteria. So again, please refer to the preprint for the breakdown of the methods and of the full results on this. So what's the secret here? What's so special about this model that it was able to replicate these trends? So in that model, the key interaction is that a decrease in bacterial growth causes a decrease in phage burst size. Now, biologically, it's something that's been suggested before and that's been seen before, and it's due to the dependence of phage replication on bacterial processes, which I've mentioned before. So simply put, what this means is that if bacteria are growing happily, then the phage can hijack their fully functioning bacterial machinery to replicate. However, if bacteria are at stationary phase, because the bacterial machinery is working more slowly, then the phage production is reduced. Mathematically in the model, this is expressed as a logistic function. So simply put, the burst size of the phage decreases the closer the bacterial population size is to the carrying capacity. And this is the exact same function that I'm using to also model bacterial growth in general in the model. So that's an interesting result because previous attempts to model this sort of link between bacterial growth and phage burst size have relied on complicated, quite abstract functions with various exponents. So here, that's the first time we've been able to link it so linearly in a way. So that's the first insight that the model is revealing here, so the invisible dynamics of phage predation. But what about transduction then? So remember I said the transducing phage are invisible in vitro, I can't count them. However, they're present in the model, they have a compartment there. So because I fitted the model again using MCMC methods, I can infer what these transducing phage numbers must look like in vitro. And this is what this looks like. So that's the phage incidence over 24 hours for each of my 3 datasets. In green are transducing phage and in gold lytic phage. And this tells me that I'm getting about 1 new transducing phage carrying an AMR gene that's being generated per 10 to the 8 lytic phage, which is consistent with previous attempts to estimate this. So again, it doesn't sound like much, 1 per 10 to the 8, and it's frequently dismissed as true to be relevant. And yet here I've shown that it's enough to consistently generate double resistant bacteria in less than 7 hours. So I'd argue that it does matter. Now, the second piece of evidence I can get from the model to tell me that transduction matters is that if you think about it, a new DRP, double resistant bacteria or double resistant progeny, can be generated either by transduction or by replication of existing DRP that have already been generated by transduction. And again, that's something we can't separate in vitro just by looking at the curve, but it's something we can look at inside the model. And if you look at the fraction of DRP generated by transduction through time and in each of the 3 datasets, you see that it's always higher than 0.5, which means that DRP are always predominantly generated by transduction, which really highlights again the importance of that mechanism. Okay, so summing things up, so we've designed a framework combining lab work and modeling to investigate the dual nature of bacteriophage, their dynamics of both phage predation and generalized transduction. In terms of rethinking AMR, so these results suggest that current models of AMR spread may underestimate potential of phage to contribute to the growing burden of AMR by not factoring in transduction. In terms of rethinking phage predation, this is another result which builds upon some experimental previously published research on the link between the phage burst size and the bacterial growth. So here, this is the first time that we've been able to link it so straightforwardly, these two components. So in terms of next steps, again, thinking about this little loop that I'm working on, it's going to be to use that model to further analyze these dynamics in the presence of antibiotics. So that's something which we'd expect to commonly see in the environment, phage, bacteria, and antibiotics together. So that's what I'm currently working on, and using the model to do this to then later generate new hypotheses to test in the lab again. Okay, so hopefully I've shown you how mathematical modeling can be useful to look at these invisible dynamics of all these invisible organisms all around us, which I find pretty cool. So thank you very much for your attention. So I'd like to thank my 2 supervisors, Gwen Knight and Jodie Lindsay, but also Aja Gupta and Jacob Wildfire. So again, preprint information here, and please don't hesitate to contact me if you have any questions about this. Thank you very much.